for today is actually precipitation. If an antibody will combine with something that is cellular, you will directly observe the reaction, then it's agglutination. So meaning, upon addition of antibody antigen, there's a gross macroscopic reaction, then that falls under agglutination. May rationale man siya kasi the antigen is cellular. Now, if you're going to compare two antigens, IgM and IgG, what is best analyzed using agglutination is IgM. Now, in terms of precipitation, the only difference between these two is that in precipitation, the antigen is soluble. So, meaning upon addition, you won't see any direct visible reaction. So, tingnan natin dito, pasensyahan nyo na yung test tube. There are the antibodies and these are the antigens. If they will combine, since they are both soluble, you will not see a direct visible reaction. But if you allow them to incubate, they will form an immune complex, and upon this one, they will lose their solubility and they will start to precipitate. And the best antibody analyzed using this one is actually IgG. So if you will be asked during the moving exam or in the lecture, precipitation tests are designed to measure the answer, IgG. Kasi kung IgM yan siya, it would forfeit the purpose. Kasi kung IgM na pentameric, automatic mag-precipitate out ang kumbaga. Kasi bakit man, you allow them to cross-link before they will precipitate. So the best antibody for this one is actually IgG. Now what you will do in experiment number 3 is that you have the agar to which the antibodies are actually suspended, and this is your well. This is where you're going to place your sample. I think the sample that we're going to look for the blood is C3, one of the complement product. Ay, immune system na kayo sa micro?
Yes.
So this was already mentioned in your lecture, right?
Yes.
So this is an antigen. This will serve as the antigen. Once you will place it on the well, since it is surrounded by antibody to C3, what happens? Antibodies are stuck within the agar. They will not move. It's the antigen that will move towards the antibody. So try to imagine C3 going here, C3 going there, and basically in all directions because they are surrounded by the antibody. So let's try to compare. After that, you allow that to incubate and you will see a precipitation ring. We will call this as precipitin ring. So if this is patient A, this is patient B, this is patient, hindi siya symmetrical, patient C. So you will have an idea that there are more antigens in patient C than B than A. But you will not observe this reaction immediately because this is actually precipitation. That's why the method is radial immunodiffusion, because the antigen diffuse radially. Tama? Tapos immuno because there's antigen and antibody, and what did the C3 do is to diffuse radial immunodiffusion. Tapos this will be thoroughly discussed in your lecture, the second experiment, the immediate versus the delayed type of hypersensitivity. But we'll just have an overview lang so that you'll know what you're doing. Immediate, basta pathophysio niya, if you are first exposed to the allergen, what your body will do is, the macrophage will present the allergen to T helper 2, T helper 2 activates B cell become plasma cells, and if this is T helper 2, B cells will most likely become IgE producing cell. So kasi kung T helper 1 yan, basta pag-discuss yun sa GenPat nyo sa immune, the B cell will prioritize forming IgG and IgM. So this is the common infection. So if you're a person born with more active T helper 2 than T helper 1, you are more allergic. If you have more active T helper 1, then you are prone to develop autoimmune disorders. That's why there must be imbalance. Anyways, the IgEs that you will produce have receptors on the surface of mast cells. And you know very well that inside these cells is histamine. But upon first exposure, the IgE will just display themselves on the surface of mast cell without any corresponding consequence. But if you're exposed for the nth time, what happens is every time you're exposed, you produce more IgGs in quantity. And if the allergen now can cross-link two IgEs at the same time, that's the time that this mast cell will be triggered to release histamine. So meaning it would take a lot of exposures before you will actually manifest the, what you call that, the allergic reaction. That's for penicillin. The other one is actually tuberculin. So what's the point? Let's say you were exposed to, what is the organism? Mycobacterium tuberculosis. So meaning the macrophage will present the bacilli to T cell, but this time it's not T helper 2 that will react, it's T helper 1. What will this T cell do is it will recruit other T cells and other macrophage. So given that you were injected with tuberculin, so meaning that's, what's that? Bovine? What's the Mycobacterium bovis? Meaning it shares the same antigen as that of tuberculosis. Meaning, if your macrophage will encounter it, and since you already have T cells that already know this antigen, what they will do? They will recruit T cells and macrophage. You imagine the area will become crowded with T cells and macrophage. The area will really become swollen. But since it would take time before they will recruit the cells needed, that's why the reaction is called delayed. That's why you will only read the result after 24 hours. While this one, you only need to cross-link the IgE. If you already have them in abundance in your body, the reaction would be very immediate. So your assignment would be, what's the first event after cross-linking of two IgE? Release of histamine. Why? Because the histamine are already stored inside the secretory granules. But there's that second event. Sige na, release of preformigator. Where are these preformigators? They are stored within the sweet tissue. Ano man ang preformigators? Ang example, histamine, heparin, cytokine, bahala kayo. Tapos number 2, ano yun? Tissue. Phospholipase. Phospholipase, what? Bago lang kasi ako nag-bio dito kaya malala pa. Phospholipase A2 will act on the cell membrane. Cell membrane, what? Release what? Arachidonic acid. Acted upon by lipo-oxygenase becomes what? Leukotrienes. So meaning, if the person is having allergic reaction, you gave antihistamine, he's not reacting, meaning the event is no longer number 1, it's number 2. Tama? Why do you consider leukotrienes as slow reacting substance of anaphylaxis? Why? Which will first cause the symptoms? Histamine. Which will act later on? Leukotrienes. Slow reacting substance of anaphylaxis, but more sustained, more immediate, more sustained. So, ano yung number 1, yung penicillin? You're testing for IgE. While number 2, you're testing for the function of your T helper 1, whether it will recruit T cells and macrophage. If you don't have TB in your body, so ano man, macrophage will still have to present it to T helper. The process is going to be very long. That's why less T cells and less macrophage. But if you were exposed previously, this one already knows the antigen, it will be activated directly. So it will what? Recruit more T cells and macrophage. That's why you will see the reaction. Ano man ang positive result nitong dalawa, the area will become swollen. Why do you think there's swelling in immediate? What's the mediator? What will it do with the blood vessel? It will vasodilate and
increase permeability.
So fluids will leak out of the blood vessel, causing the area to become swollen. Pareha lang. O yun lang yung ginagawa nyo today. Okay.
Okay, Doc.